Good morning, everyone. My name is Joyce Coleman. I'm the Executive Director of the Vasculitis Foundation, and I'd like to welcome you to today's webinar. Today's webinar is the genetics of vasculitis and current research. Our presenter is Dr. Amar Sawalha, the Professor of Internal Medicine and Professor and the Marvin and Betty Danto Research Professor of Connective Tissue Research at the University of Michigan in the Department of Internal Medicine in the Division of Rheumatology in Ann Arbor, Michigan. His research focuses on the genetics and epigenetics of complex autoimmune and inflammatory diseases, including lupus and systemic vasculitis. Dr. Sawalha has built a unique investigative consortium that includes physicians and researchers in Turkey, Italy, Germany, the Netherlands, Japan, and Korea to study the genetic contribution to Behcet's disease and Takayasu's arteritis. In 2009, the Vasculitis Foundation funded Dr. Sawalha's study, Identification of Susceptibility Genes in Takayasu's. During today's webinar, if you have any questions, please submit them through the chat window on the right-hand screen of your window, and we will be doing a Q&A after Dr. Sawalha's talk. Kaylin Young from the VPPRN network will be coordinating the questions along with Ed Becker. So I'd like to welcome Dr. Sawalha to the webinar. You have the floor, Dr. Sawalha.
Thank you, Joyce. It's my pleasure to be presenting today and talk a little about our work and what is known regarding the genetics of systemic vasculitis. And so in diseases that we study, try to advance the slide here. Vasculitis is what we call a complex disease when it comes to etiology. And the reason why we say it's a complex disease is because it's not a purely genetic disease and not a purely environmental disease. It's a disease that can be caused by interaction between genetic elements and the environment. And so if you look at diseases in general and look at the contribution of environmental factors versus genetic factors, you'll find some diseases or conditions that are caused entirely by the environment, and then diseases that are entirely caused by genetic factors. Systemic vasculitis is kind of in the middle zone here, where we have contribution from genetic factors associated with environmental factors. And the assumption is if you have more genetic risk for the disease, then you'll require less environmental triggers to develop the disease compared to people who have less genetic risk, who will then require to have a higher environmental contribution to get the same phenotype, or in this case vasculitis. So to introduce genetics, what are we talking about when we say genes or genetics? What we are talking about is the nuclear material here represented by this picture showing the nucleus and then showing these chromosomes that include all the genes or the genetic codes in our body that dictates what the cell can do and function and how we can perform all these different biological processes in our body. So everything is really encoded within what we call the genome, or our nucleic acid code here. Just a few terms that we'll be using throughout the talk and in genetics in general. When we say a gene, we're really referring to a fragment or a segment of the DNA that tells the cell what specific protein to make. So our cells make a lot of proteins to be able to function. And each of these proteins is encoded by a specific gene or a specific fragment of DNA. Now, these genes can be different between individuals, and this variation between individuals within a gene we call alleles. So humans are different, and we don't look all exactly the same. We have differences in our phenotype. We have differences in, let's say, for example, hair color, skin color, height, weight, and so on. And all of this is dictated by genes. Now, a variation within these genes is what really makes us different and this variation is what we call alleles. And so some of us, when it comes to diseases, might have a variation of a gene that is more likely to cause a specific disease, and some of us might have another variation of that same gene that will make us less likely to get a disease, and so on and so forth. And that's what we really are looking to study when we study the genetics of diseases. When we refer to the genome, what we're really talking about is the entire repertoire of genes that we have in the body. So when we say a genome-wide association study, what we mean is studying the entire genetic background of an individual, and specifically in this case looking at patients with specific diseases compared to healthy controls. So all of these genetic material, it's very complex. It's compacted actually in chromosomes like this, and although it's very complex, at the same time it's formed by a combination of 4 basic nucleotides that are depicted here. So it's basically the sequence and the variation within these nucleotides within the sequence is what causes the genes to be different from one individual to the other. And so this genetic material, as I mentioned, is packed into chromosomes. In human, we have 23 pairs of chromosomes that are shown here, and then these are 22 what we call autosomal chromosomes present in both men and women, and then we have the sex chromosomes. In this case, we have either an X chromosome and a Y chromosome, which makes this individual a male, or we have 2 X chromosomes, which will make this individual a female. And so when we talk about genetics, we have to remember that modern genetics really started by Gregor Mendel's work. He was a monk, Augustinian monk, who lived in the 1800s, and he basically observed differences in the phenotype of peas flower colors. And so he was able to, using these observations between pink versus white color, to make specific contribution to genetics, which we actually still use to date, which is called Mendel's laws of inheritance. And so vasculitis is not a Mendelian disease. It's not something that is simple where you have a gene that gives you a disease. So one gene gives you one disease. This is what we call Mendelian genetics. And this is an example of such Mendelian genetics. This is, for example, a sickle cell disease family. This is the father, this is the mother, this is the here a son. This is another family, and you can see in this disease, for example, having one copy of the disease gene in black and the healthy copy in white in one of the parents, then another copy from the other parent, in this case the mother of these 2 childs here. You can see that if both of these copies end up in one of their children, which in this case is their son, then this person will get the disease. And this is what we call an autosomal recessive pattern of inheritance. Very simple genetics. You have one amino acid change, in this case in the hemoglobin gene, that gives you, instead of hemoglobin A or the normal hemoglobin, what is called hemoglobin S or sickle cell hemoglobin. And then this person, when they get this 2 copies of these disease genes, they will develop the disease. And so one gene, one disease, but this is really not what we're talking about when we study vasculitis. Vasculitis genetics is what we call complex, so it's not really Mendelian. It doesn't follow the Mendel laws of genetics. And so what that entails is that we have a number of genes. So we have probably in the order of tens or maybe hundreds of genes that might contribute to the genetic risk for getting a systemic vasculitis. And so these genes, together with environmental factors, some environmental factors such as an infection have been reported in some vasculitides. Some medications have also been shown to increase the risk of vasculitis, but also because of that we know that there are some environmental factors that are not necessarily drugs or medications that have been observed to increase the risk for developing systemic vasculitis. For example, silica exposure has a strong link to be associated with ANCA-associated vasculitis and so on. So these environmental factors then, along with the genetic factors, they produce an altered immune system response that ultimately leads to a disease. So we start with a genetic background. Again, tens or hundreds of genes, each contributes very little to the risk of the disease. And then on top of that, we need some environmental factors. Some of them we know of, some of them we largely still don't know. Together, they will cause an alteration in the immune system, and then the disease will happen. And so recently, we have been able to study genetics much more robustly than we were able to do in the past. And part of the reason is completing the Human Genome Project by the NIH. And so that project spanned for about 13 years, really showed us how the genes, what the entire sequence of the genes in the human genome look like. And so when we know the normal sequence of the genes, then we can study variation in the genome. We were able to then, through a HapMap project, which is still ongoing, identify these types of genetic variations between individuals. So again, humans are outbred in a sense. We are admixed. We have a variety of alleles, a number of variants that are different between different individuals. So to study these variants, we need to know the normal distribution between populations of these genetic variants. And so once we know the normal variation, then we can also study the genetic variation in disease and see which of these variants are associated with vasculitis. And so there are also advancements in the way we can do a genome-wide, again, looking at the entire genome and the human genome-wide association studies, and that can help us look at now at about 5 million genetic variants at the same time with a simple chip. So we start with a DNA sample, can be a saliva sample, it can be a blood sample, and then a simple chip, do some genotyping, we can identify pretty much almost all genetic variation in the individual that we study. And so now we can also sequence the entire genome, and we can get to understand more variation by doing sequencing. We have sequencing data from about 27 populations to date, almost 4,000 individuals sequenced. So again, we are identifying the repertoire of genetic variation that are present in normal, which will help us identify disease variants. These are getting cheaper with time. We are now able to get the entire sequence of the entire genome for about $1,000 or so. And then obviously we have a lot of advancement in bioinformatics and computing technology to be able to allow us to analyze these complex data sets. And so when we look at these numbers of GWAS studies reported, we can see an exponential increase in the gain of knowledge in genetics in diseases in general, as you can see here. You can also see categories of diseases, and each dot represents here a category of disease and a genetic variant that was found in these different chromosomes. And so you can see we have now, this was until 2013, now we have over 2,000 GWAS studies published in different diseases. So whereas vasculitis? Vasculitis a little bit lagged behind many of our diseases that we studied in genetics, and part of the reason was because vasculitis is rare, relatively speaking, compared to other autoimmune diseases. And so in order to be able to find genetic variation in genetic studies, we need a large sample size. So it took us a while to really assemble cohorts and patient samples large enough to be able to study the genetics of vasculitis. And so if one wants to summarize the genetics of vasculitis, this is a recent paper we just published, and these are here 4 types of major vasculitides: the ANCA-associated vasculitis, Kawasaki, Takayasu arteritis, and Behcet's disease. These are the number of genetic variants in each of these diseases depicted here that we feel comfortable about at this time. What is important to see here is that there is no overlap between genetic variants for Behcet's disease, for example, and genetic variants that cause Takayasu arteritis, or between Takayasu and Kawasaki or ANCA-associated vasculitis and any of these other vasculitides. So it seems that the genetic risk for systemic vasculitis is specific to the specific vasculitis subtype. What do we know so far? ANCA-associated vasculitis was studied a lot in recent years in terms of looking at genome-wide association studies. Again, there were two landmark papers. The main paper really came out of the group in the UK in 2012, and what we learned is not only that we have specific genetic variants for ANCA-associated vasculitis, such as in the region called HLA, which really plays an important role in how our immune cells recognize foreign antigens. Also proteinase 3, which many of you know that patients with ANCA-associated vasculitis, especially C-ANCA vasculitis, they have antibodies against proteinase 3, so the gene that encodes this enzyme actually is a risk factor for the disease. Alpha-1 antitrypsin, which actually inhibits proteinase 3, was also a risk factor, and so on. But we also learned that the C-ANCA vasculitis and the P-ANCA vasculitis are genetically very distinct. In fact, the genetics of these diseases are more likely to distinguish between the C-ANCA and the P-ANCA vasculitis than the clinical syndromes, the GPA and the MPA that is associated with the antibodies. So genetics can also help us classify the diseases a little more, and that's one other thing we like to, or we aim at getting from genetic studies. In Behcet's disease, we and others were involved in again, genetic studies in this disease, and we're starting to understand that it's not only the HLA that is associated with the disease. We have other targets now that we can develop for future therapeutics, or also help study to help us understand the disease better. Some inflammatory genes like the IL-10, the IL-23 receptor, which is involved in specific cell types that are called T cells, specifically what we call Th1 cell, and also a Th17 cell. And then again, some signaling molecules important in specific immune regulatory functions that we hope we can target for future therapeutics in the future. And so the question that always comes up when we talk about genetics is, what is the risk of a family member to get vasculitis if I have the disease? And that's a very important question. Now, remember that vasculitis is not a simple genetic disease. It's a complex genetics disease, meaning that we have multiple genes, each contribute a little bit to the increased risk of the disease. So it is not, in a sense, a genetic or an inherited disease, but it does have a genetic contribution to its pathogenesis or to its etiology. And so if we want to talk about the risk of a family member to get vasculitis, there are two important factors that help us estimate this likelihood or the overall risk. First, we have to know what's the incidence of the disease overall in the population. And second, we need to know what is the relative risk for getting the disease if you have a family member that has a disease compared to the general population. So let me give you an example of this. So if we look at GPA, for example, formerly known as Wagner's granulomatosis, study was done in Sweden. Now GPA has a prevalence that tends to be higher in Northern European populations and less in Southern European populations, opposite to the to microscopic cholangitis, which tends to be more common in Southern Europe, for example, compared to Northern Europe. But in this example, this is looking at GPA in Sweden. In this study, there was an estimated population prevalence, in this case about 1 in 100,000. So remember, vasculitis in general is rare, and this is actually one of the more common vasculitides. Takayasu, for example, has a prevalence of of 3 per million, and so on. So in this population, the relative increase in risk in a child of a patient with GPA was 2.56 folds compared to the general population. So this seems to be that you increase the risk by 2.56 times the general population. But remember, the overall prevalence of the disease is 1 per 100,000. So when we do the math for this, the incidence of GPA in a child of a patient is actually 2.56 times 1 in 100,000, which is still 2.56 per 100,000, which is still very low. And so in other words, the risk of transmitting, or the risk that your child will get the disease because you have the disease, is very, very low. And this is very important to clarify. So why do we study the genetics in vasculitis? I alluded to some of this before. One main reason is to understand the mechanisms of the disease, because when we find the genes that contribute to the risk of vasculitis, we can understand what specific immune cells, for example, are important in causing vasculitis in general, or if we look at more specific individuals with specific subphenotypes, so patients with kidney involvement in ANCA-associated vasculitis, for example, compared to patients who don't have kidney involvement, then we can know what might be contributing to the kidney involvement specifically in vasculitis, or the skin, or the brain involvement, or eye disease, for example, in Behcet's and other vasculitides. So we want to understand the mechanism of disease and also the mechanism of the subphenotypes, or the organ involvement, by studying genetics. We can also understand something about prognosis. For example, if we identify a genetic variant that makes people that have this variant more likely to get kidney disease, we might be able to prevent this from happening by doing more frequent monitoring for the kidneys, for example, and so on. So we really hope that we can get this information to the clinic and helping us stratifying how we can monitor patients. Again, by identifying specific immune molecules that are involved by genetic risk, we can target them specifically for therapeutics, potentially novel therapeutics that are less toxic than immunosuppressives that we currently use in vasculitis. And then patients might respond differently to different medications. And that's what we call pharmacogenomics, or individualized medicine in this case. So perhaps people who have risk variants in one specific molecule will benefit from a drug that targets this molecule, whereas individuals who don't have this risk factor but still have the disease might benefit from a different medication. So we can hope to use this information to individualize therapy in a pharmacogenomics approach. So we have interest in Michigan in studying vasculitis and specifically the genetics of vasculitis for some time. I'll share with you some of the studies we've done. Of course, we cannot do any of these studies in a rare disease like vasculitis without a large sample size, and that requires a collaboration really between us here within the US, North America, and in our case, worldwide collaboration really. Many countries are involved and continue to be involved in these studies to be able to do this. So this is an example of a study looking at Takayasu arteritis. So when we first started examining Takayasu and looking at its genetics, the only risk factor that was known is within this HLA molecule that again dictates how cells interact with foreign antigens. But when we started doing more detailed studies, we were able to identify new targets in specific molecules like the Fc gamma receptor, which is again a receptor that perhaps we can inhibit or interfere with in the clinic. A target that is involved in specific immune cells. In fact, there is a biologic treatment now approved for treating another disease that targets this molecule. So perhaps this treatment can be explored further in Takayasu arteritis based on these findings. More recently, we did a genome-wide association study again in Takayasu, We found another inflammatory molecule, which is IL-6. There's a medication called tocilizumab that seems to be successful in treating a disease that is closely related to Takayasu called Giant Cell Arteritis. And so perhaps using tocilizumab in clinical trials in Takayasu is warranted. And so this is the type of information we hope to get, and we can't get that without doing large studies that include a large number of patients and a extensive collaborative effort throughout the entire world, really. In Behcet's disease, we learned that the HLA is more complex than we think. The HLA association, in fact, is 4 different associations at least, and it might involve regulation of the gene expression, not just changing the protein itself, but regulating how these genes are expressed might be involved in the risk of Behcet's disease, and we're working and trying to elucidate more mechanisms in this regard. And so the other aspect of genetics that we are interested in is called epigenetics. So in addition to looking at changes in genetic sequence, which we call genetics, we can also study how these genes are regulated or expressed, and this is what we call epigenetics. And so this basically studies how these genes are expressed in the cells and what regulates their expression. So one mechanism we study is called DNA methylation. And so genes that are regulated by methylation, when they're methylated, represented here by black circles, the expression of the gene is suppressed. So you can imagine if these genes are not methylated, and let's say they are abnormally not methylated, then you have more gene expression. If this gene is pathogenic, you have more expression of a gene that can induce disease or induce immune activation, you can get a disease phenotype. If you suppress the gene again abnormally by methylation, and the gene is important to prevent an immune response, then you can see how we can have an overall activated immune response. So it is very important to also think about epigenetics. This is an example of a study, some data from Behcet's disease again. And so what we found here is that we found a number of genes that are involved in the cytoskeleton or involved in how cells interact with one another that are abnormally methylated in Behcet's disease. But what was interesting to us, and this is in a specific cell type we looked at, is that when we looked at patients compared to controls and looked at these methylation changes, and then looked at these same patients, compared them when they became in remission or after they were treated for the disease, we could demonstrate that some of these abnormal methylation changes were completely, or in some cases almost completely, reversed. So those that were lower in patients compared to controls become high in patients when they're in remission compared to patients who are in active disease, and vice versa. So what this tells us is that we have some sort of dynamic change in some of these genes that we can perhaps target for therapy. So one of the main themes that came from these studies, again, is genes that regulate cytoskeleton. These are the genes that regulate how the cells move and how the cells interact with one another. And so these genes seem to be more active in patients with Behcet's disease, suggesting that the cells can interact more. These are immune cells, and so if they interact more, that means more immune activation. If the cells can migrate by making these extensions, migrating to the tissue, invade the tissue, and cause more inflammation, and this process is increased in Behcet's disease based on our findings. Then the next logical step, of course, since we also saw that these changes are reversed when patients are in remission, is to develop inhibitors for these specific molecules. And so we're working with one target that is involved in making these extensions here, these extensions, and this target, we have an inhibitor for it, and we can demonstrate in tissue culture that we can inhibit the cell migration by inhibiting this target. So potentially we can develop new treatments by targeting these new molecules we learn about through our epigenetic studies and through the genetic studies. The other thing we can do is to understand the nature of inflammation by looking at the tissues. So using genetic approaches, if we take a tissue, this is an example of a temporal artery biopsy, patients with giant cell arteritis, they typically have headaches, they have vision changes, they have jaw claudications, they have overall tiredness, pain in their hip areas, shoulder areas, and so on. Many times to confirm the diagnosis, we take a biopsy from the temporal artery. And when we look at it under the microscope, there's a lot of inflammation in this artery. Now, what we can do, and what we actually just recently did, is take this biopsy material, extract the DNA from the biopsy or from this inflammation, and use the pattern of gene expression changes to kind of know and infer what type of inflammation specifically happens in these arteries, hoping that we'll find novel treatment targets for this disease. And so this is what we recently did also and published. We did this in 12 patients and 12 normal people who did not have giant cell arteritis on biopsy. And then we compared their DNA methylation patterns, that gene expression regulation pattern that I mentioned before, between patients and controls. And so what we were able to find is some novel targets that we then were able to confirm at the protein level. So this is an example of what we found. So we found, for example, that there's dysregulation of this important molecule that is involved in T cell activation. So there's this brown staining here refers to the protein. So we after we found it by methylation studies, we then stained for it in the tissue biopsies from patients. So there's a lot of this molecules abnormally produced in these inflammatory cells within the giant cell arteritis biopsies. By the way, we call it giant cell arteritis because in the biopsy we see giant cells. This is an example of a giant cell, a cell that has so many nuclei in it. It's a classic example. So then one logical approach is that targeting NFAT in this case, because it's increased and activated in patients with GCA, might be a novel therapeutic option for GCA, and we plan to do a small clinical trial to look at some of these NFAT inhibitors. We have another molecule called CD40 ligand. Again, it is specifically expressed in T cells and can be also therapeutically targeted. There's other inflammatory cytokines. This IL-21 is increased. Its receptor also is increased. So again, these types of studies will help us identify targets that we hope we can take to the clinic in the form of clinical trials in the future. And so to conclude and leave plenty of time for questions, it's important to emphasize that both genetic and environmental factors are involved in causing vasculitis. It's not a genetic disease per se or environmental disease per se. It's the combination of both genetic factors and environmental triggers that cause the disease. We're understanding the genetic component more. We have some data on environmental factors. We're still lagging a little bit behind on environmental triggers, not only in vasculitis, but all autoimmune diseases, because studying the environment is not very easy. It's a complex thing to do, but we need to understand this component too, and then once we understand the genetics completely, the environment completely, study how they interact with one another to cause the disease. So if we uncover the genetics and show which genes specifically are involved in what specific type of vasculitis, we can better understand how the disease develops. We can identify new targets, new specific targets for therapy. Now, when we develop specific targets and specific treatments, remember that the side effect profile is expected to be much less than, for example, using cyclophosphamide or methotrexate or azathioprine, which suppresses many molecules and many cells within the immune system. So that's the hope, is to be able to have specific treatment targets that will have less side effects. Now, once we understand the genetic variation between individuals within the disease based on the subphenotype, meaning patients with GPA who have kidney disease versus patients who don't have kidney disease, patients who have more lung disease versus patients who don't have much of a lung disease, then we can also make specific treatments that will work specifically for specific groups of patients based on their phenotype. And then also, based on the genetic profile, we hope to be able to predict which organ will be affected in these patients, and therefore we can try to prevent by more frequent monitoring, more aggressive monitoring, and so on in specific individuals. And very important take-home message here is the risk of vasculitis remains very low in your family members, as I demonstrated with the example I showed you in GPA. The disease is very rare, and genetics, if you have the disease, is not going to significantly increase the risk of your siblings or of your children of getting vasculitis. And so I would like to thank you for your attention, thank the Vasculitis Foundation for all the great work they do to get us together and support research, support disease groups and awareness for these rare diseases. And I would like to thank the University of Michigan, who actually help us fund the work that we do in vasculitis. And so thank you very much, and I'll be happy to address your questions.
Great. Thank you very much, Dr. Swahala. I'll turn the floor over to Kaylin Young. Kaylin is our network manager for the Vasculitis Patient Powered Research Network, the VPPRN. So Kaylin, are you on board?
Yes, I sure am, Joyce. Thank you. And thank you to those who have submitted your questions via the chat box. Again, if you have additional questions, you can go ahead and chat those in your navigation bar. There is a little tab where you can submit questions, and I will receive them and be able to pass them on. So one of the first questions we received, and I know that Dr. Swahala, you addressed this, but this question is in regards to family genetics of vasculitis. And the question is, has there been any studies regarding different types of vasculitis diseases in the same family? They cited an example of the slide you showed for GPA specifically that talked about the slight increase of risk to children if the parent also has GPA. So do you have more information about research that's being done on different types of vasculitis in the same family?
Yeah, you know, so in general, when we look at systemic vasculitis, and in fact all types of vasculitis, what we call sporadic cases, it is much more common than familial cases. So the vast majority of patients will actually have the sporadic disease, meaning that they will not have a family member affected. However, we do have families where we have clustering of the disease. And the clustering, or the amount of clustering, or the amount of having familial disease is really very low. It has been studied in GPA, as I showed you before. Now we can use that same data to look at other vasculitis. So other vasculitides are less common. For example, think about Takayasu arteritis. No, we don't have the same type of data as we have in GPA, but let's assume we have, instead of 2.56-fold, let's assume we have 10-fold increase in the risk of getting Takayasu in your children. And so even with that, so we have a disease that have a prevalence of 3 per million, so it becomes 30 per million, and that is really still very rare. Now, we do have other studies in Behcet's disease, also in familial cases. There is increased risk in family members, similar to other diseases. These studies have been done in Turkey primarily, but again, the familial, the likelihood of getting, a family member of getting the disease remains very rare. So it is something we study. We do look for these families because they can inform us about the genetics, especially if we can get the parents as well as the affected child, but we don't find them very often.
Great, thank you. So it kind of leads into another question that was submitted about research. So you'd mentioned needing a large cohort of patients to participate in research. And when you do genetic research internationally, is this more difficult to organize than other research due to the nature of genetics? So for example, is it more difficult to get IRB approval for genetic research?
I wouldn't specifically say it's more difficult, but it is very much regulated because of the sensitivity of the issue. So we have to, the regulatory bodies within the U.S. And also internationally, they require specific assurances that have to be agreed upon before starting genetic studies. This is something that the NIH also enforces very much in their grant applications and also in the process that we have to go through, even to access publicly available genetics data through the NIH. So we have a lot of regulations to go through, but I wouldn't say that it hinders the research, but it's very important that these things are regulated because you can learn a lot about an individual through their genetics. We have not had issues with international collaborators. There are some countries that do not allow transferring genetic material from these countries outside, and that has been a small issue for us, but we're working with genotyping facilities within these countries where we can do the genotyping there and then we can analyze the data here in the US.
Great, thank you. So this is another question to our kind of related, but we received this question a lot on our social media pages, and the Vasculitis Foundation in particular receives this question quite often. Is stress a likely environmental trigger in vasculitis, and what research is kind of being done with regards to stress and vasculitis, if any?
So stress in general, can be a trigger for many diseases. Stress does affect our immune response. And studies that have been done looking at what we call the neuroendocrine disturbances that happen with stress, when you look at individuals with chronic stress, you actually see a reduction in the useful immune response, meaning that people under stress will develop, are more likely to get more infections and so on. Now, there are no specific studies in vasculitis that tell us that stress or how stress really works in the pathogenesis of vasculitis. I can tell you from my own clinical observation that I do see stress can induce flares in some of these diseases. Specifically, I see many Behcet's disease patients here at the University of Michigan, and I see from my own practice a clear indication that stress increases mucocutaneous involvement or oral ulcer, specifically in patients with Behcet's disease, and I commonly talk about this to patients. But definitely this is something that needs to be worked on a little bit more. The problem is that it is not very easy to study. There's different degrees of stress, different types of stressors, acute versus chronic stress. But I think it's healthy for any of us, whether we have vasculitis or not, to try to reduce the amount of stress, because it does seem on the chronic, with chronic stress, we do have reduction in our useful immune response that targets infections, and many of our patients are on immunosuppressants, and you don't want that increased risk of infection. So it's very important to reduce stress.
Thank you. So this is another question submitted. Does the low incidence of genetic transfer vasculitis within the family suggest that epigenetic and environmental factors may be more important than genetic inheritance?
Oh, this is a great question, actually. Yes, it does suggest, I wouldn't want to say more or less important, but I would say that it is extremely important that we understand the environment and the epigenetics. What it tells us is that the genetics do not, on its own, explain the disease. And in some diseases, I would expect that the environment might be more important. In some diseases, maybe the genetics will be more important. And then it also depends on individuals. So the first slide, as I showed, I think I like the idea of genetic epigenetic environmental interaction. And I personally think, and we've seen examples from other diseases, for example, lupus, is when you have more genetic risk, then you don't need that much of environmental triggers to get the disease. If you have less genetic risk, you need much more environmental triggers to get the disease. So it's really kind of a scale, how much genetic risk versus environmental triggers you have. But yeah, this is a great question, and it does suggest that environment and epigenetics play a very important role, and we need to study this more.
Great. Thank you. And so kind of related to the same question, there was another submitted about, first being diagnosed in 2007, when there wasn't a lot of solid research that this particular patient was aware of that had been completed regarding known causes. So infection, genetics, and environmental causes were the top three being discussed. So in your research, you discussed environmental as well as genetics. Are there any supporting evidence that a major or acute infection causes something to trigger the vasculitis?
Yeah, I mean there is evidence for infections to trigger vasculitis in GPA, for example. We have evidence that some of the bacterial infections might have antigens that cross-react with the main target for the antibody response in GPA, which is the proteinase 3. So there is evidence for that. There's also evidence for parvovirus infection being involved. But the studies are not conclusive at this point in this regard. There need to be more work done. Overall, we do think of an infection as a trigger for a flare of diseases in autoimmunity in general, and that's different from causing the disease at the offset. But patients who have a systemic autoimmune process, when they get an infection, they're more likely to flare, and so that suggests to us that you might have some cross-reactivity between the infection itself and the targets of the immune response in vasculitis. Another thing is that when we get an infection, we get a lot of oxidative stress. We get a lot of internal stress within the cells that try to get rid of this infection. And so when you trigger an immune response in that way, you can also inadvertently trigger the immune response that causes the vasculitis. But we don't have, in terms of causation, we still lack a conclusive evidence that links a specific infection to a specific disease. We have evidence, but it's not very conclusive.
Okay, thank you. Ed, I know that you had received a couple of different questions. Would you like to pose some of those now?
Yeah, sure. Thanks, Kaylin. So I did get quite a number of questions, but I'm going to zero in on just a couple of them. And this one is actually two different questions, but I think with the same theme and idea. This woman wrote, "My husband and I want to start a family, but I'm so afraid that I will pass on my polyarteritis nodosa to my child. Is this a valid fear?" So that's the one question, but I think the second question that was asked relates to that. And someone wrote that they understand that there's a very low risk that they would pass along the same vasculitic disease they have to their children, but that they have heard and wondered what your thoughts were, that there still might be a greater risk for some type of autoimmune condition, probably not vasculitis, but could the kids be at risk of some type of an autoimmune illness or condition?
Mm-hmm. Yeah, obviously the answer to the first question is the risk is very low, and I wouldn't really get concerned so much about the possibility of transmitting the disease. It's not a genetic disease, there's genetic contribution, and the risk overall is low to have a child with the disease if you have the disease. Now, in terms of other autoimmune conditions, we do see some what we call clustering of autoimmunity within families, And the reason for that is that some of the genetic risk loci you're identifying are actually common between diseases. Now, PAN is not one of these diseases I think of in that way. It is usually an autoimmune mediated disease with an antibody, for example, lupus, thyroid disease, rheumatoid arthritis, Sjogren's. They do tend to cluster, sometimes within families. I don't think of vasculitis the same way. I don't think we have data to conclusively say this, but in PAN, it's not an antibody-mediated disease. I would not think there's a high risk of getting another autoimmune disease within these families, but theoretically, there is an increased possibility of autoimmunity. But if that is the case, I would assume it to be very, very low.
Okay, thank you. Another question someone asked is it possible that some of our grandparents or distant relatives from years ago may have had vasculitis, but it was misdiagnosed at that time just because there wasn't enough knowledge about the disease at that time?
Yes, that's very possible. That sometimes explains when you look at incidence and prevalence studies in diseases across a period of time in different parts of the world, you will see sometimes variation, and part of that is because of not recognizing the disease or lacking the facilities to make a diagnosis, or lack of medical care, and so on and so forth. So yes, it is very possible that we had patients who in the past had vasculitis that we did not recognize.
Okay. And last question. When you say that drugs are a possible factor in getting vasculitis, what does that mean? Are you talking about a reaction, the body's reaction to a particular drug?
Yeah, there are some drugs that have been shown to induce antibodies that we see in ANCA-associated vasculitis, for example. Some blood pressure medications that we don't commonly use, such as hydralazine, has been shown to cause a drug-associated ANCA vasculitis. Minocycline is an antibiotic that's not very commonly used, that sometimes has been also linked to developing vasculitis. Propylthiouracil, a medication, again, used to treat hyperthyroidism. This is not commonly used at all, has been linked. So there are a few medications that can be associated with the development of autoantibodies we see in vasculitis, and in some cases even clinical syndromes or clinical vasculitis. Now, the good news is that these medications, when they're discontinued, in the vast majority of cases, the vasculitis goes away. But the importance of this finding is not really these specific drugs. It just tells us conceptually that it is possible that we have other triggers in the environment that we don't know of that can work in a similar way.
Okay, thank you. Kaylin, I'll turn this back to you.
Perfect, thank you, Ed. So there were several questions that were submitted via our chat box that asked similar questions to the ones that Ed just posed with regards to having multiple autoimmune diseases within a family or multiple manifestations of rare disease within one family, whether that be a parent or aunts and uncles. And so I think that the general question, and I know you've kind of addressed this, but the general question I see emerging as a theme is there an interrelatedness between autoimmune diseases or rare diseases writ large in a family and passing that along? So maybe not a specific disease or not even a specific type of vasculitis as much as lupus or an entire range of other autoimmune diseases.
Yeah, I mean, as I mentioned, there is this slight risk. We don't see it much with the vasculitis spectrum of diseases. I see more, for example, of that between lupus and Sjogren's, or rheumatoid arthritis and lupus, and so on. Overall, I think it is not something that we see much of. I realize there is a slight increased risk within family members, as I, showed you the data in GPA. But if you look at this overall risk and likelihood in specific family, is still very, very low.
So
in other words, it's more likely to get the disease by chance than to get it because your family member had the disease.
Okay. Great, thank you for clarifying. So this question is actually about medications and their effect on the genetic base of the disease. So the question is, the treatments today for GPA specifically, like Rituxan, cyclophosphamide, prednisone, etc., that are being used to treat the disease, how is that affecting and or treating the genetic base of the disease? Or is it more trying to reprogram certain aspects of the gene?
Yes, so the medications we currently have and we currently use for the disease are largely non-specific. And we're not really altering any of the genes or specifically a gene product by these medications. We're pretty much suppressing the entire immune system, for example, with cyclophosphamide. Now, in the case with rituximab, it is targeting B cells, so we're depleting, completely depleting a population of immune cells, and that seems to work very well. It is more tolerated than cyclophosphamide because we're targeting a specific cell subset, but still we're not really specifically targeting a molecule. That might be a way to have medications that are more effective and at the same time more well tolerated. But we are not really altering the genetics in any of these medications.
Thank you so much. So we are almost at the top of the hour. So I would like to add one last question, and then thank you for your time. But are there opportunities for patients to contribute to future studies regarding genetics? So especially if you have multiple occurrences of vasculitis in the same family, or just want to participate in research to help contribute to that, to the data that's so needed for this research to happen.
Absolutely. We are always in need for more patients to study because, as I mentioned earlier, the disease is rare, and the more patients we get an opportunity to study, the more we can learn about these diseases. And so I would encourage patients to contact their physicians and maybe if the Vasculitis Foundation is also willing, maybe contact the Vasculitis Foundation with a specific research interest, and we will enroll them in the specific studies that we are doing. We are very interested in the genetics. We're collecting samples from various types of vasculitides for our genetic studies. We're also collaborating with the Vasculitis Clinical Research Consortium, the VCRC, that also has a mechanism to collect these samples and have them sent to their laboratories for preparation, and then the DNA gets sent to us for these very specific studies. So absolutely, I would encourage participation in all studies in vasculitis.
Great, thank you so much. Joyce, I'm going to go ahead and pass it to you as we close out.
Okay, great. Thank you, Kaylin and Ed, for moderating the questions. Dr. Swahala, thank you so much for giving this interesting presentation, and thank you for all your research efforts. And I know you also take care of patients, so thank you very much for all your work in the vasculitis world.
Thank you very much, Joyce. It was my pleasure.
Great. And I'd also like to thank all of the attendees who participated today. We did record the webinar, and it will be placed in our library on the VF website once we edit it. And I would just like to say thank you all for attending, and have a great day. Happy Wednesday, right? Hump day, right? Thank you.
Thanks.